So we move to next talk. Doctor Charalampos from Edinburgh Napier University. His topic is Synaptome Explorer, a tool for real-time visualization of the mouse brain at a single synapse resolution. Welcome.
Yeah. Thank you very much. Right, hello, everybody. So I'm going to talk about a tool that I developed to help visualize the mouse brain or mouse brain sections at single synapse resolution. So while I'm currently with Edinburgh Napier University, this was done while working with the University of Edinburgh at the Centre of Clinical Brain Sciences at the lab of Professor Seth Grant. And this work was funded, among other work, by the Wellcome Trust. So the overall problem domain is the lab's aim was to map the synaptome, to figure out the synapses, what they do, and how they are organized. And you can find some related publications at the bottom if you want to check out further. I will focus more on the software side rather than on the more neuroscience side. So they applied genetic tagging on the mice. They applied postsynaptic proteins, PSD95 and SAP102. And then they sacrificed the mice, and they got some tissues, and they imaged them using spinning disk confocal microscopes. And the output of that process was a large image data set. So it was the entire brain section at single synapse resolution. And these raw images were passed into an image analysis pipeline that identified the synaptic puncta and their properties. And also the scientists had to delineate the different regions of the brain because mouse brains and all brains are different to each other. So, for example, to figure out where the hippocampus is or where the cortical plate is, they had to point it out where it is in the images. So we ended up with a big data set. And the scientists typically used MATLAB or Python or ImageJ or Fiji to work with this data. My part here was bring all this data together and put them all in a real-time visualization tool to be able to very conveniently see everything together in real time. So I'll first focus on the microscope images. So this example image shows a sagittal section of an adult mouse. So this one, for example, was 325 by 150 tile images. Each tile was 512 by 512 pixels. And this means we ended up with 50,000 tiles, or 50,000 images. And the format for that was a 16-bit TIFF file, and it was a single channel file. So we had one set of files for PSD95 and one set for SAP102 because it was different imaging sessions. So we ended up with 100,000 images. And the total data size for that ended up being about 50 gigabytes. So one of the goals of this was to be able to explore this data set in real time. So very fast. Like you move around with a map on Google Maps, the same way to do it with a mouse brain. So one of the techniques that we used for that was GPU-friendly block compression. So block compression is a type of compression that, while it doesn't give us a high compression ratio, what it allows is to have this data, this compressed data, to send directly to the graphics card, and the graphics card is able to decode it on the fly. So there are several block compression formats. The one that we used in the end, and the choice depends on the hardware platform. For example, if you're running it on a desktop computer or on a mobile platform or on the web. So for our case, it was run on a desktop computer, and we have two channels, one for PSD95 and one for SAP102. So we use the format called BC5, which compresses 4x4 block of pixels into 16 bytes. So the size now from 50 gigabytes, now it has been compressed to 13 gigabytes, which is much better, but still it's a bit too much to load into a regular GPU. So the step afterwards is to use a a technology from modern graphics APIs, which is sparse virtual textures. And what this is, it's a type of image that we can have a very, very big image where its data doesn't have to be all loaded in memory at the same time. So some areas can be empty, and while we're seeing different areas of the image and we try to focus on some, then we can load the data there dynamically. So this is very convenient because the data for the entire image might not fit in memory, which is the case with our microscope data. So we use this technology, and as I said earlier, we have about 50,000 tiles, but not all 50,000 tiles contain useful data because some of the tiles fall completely outside the mouse spring. So we don't take this into account. So the useful tiles, we compress them with a GPU block compression format, and we pack everything into a big data buffer. And we calculate the mapping between the different areas of the big image to our big buffer in memory. So this is all done offline. So while we're running the real-time visualization program, we just map this special image into a rectangle, on the big rectangle, and then we just render it. So when we are moving our view while using the application, depending on the area that we're looking at, we just make sure that we have that data in memory. So we dynamically load and unload memory as needed. So this ends up being very efficient and allows us to have this sort of Google Maps style visualization you can see here. So here is like a zoomed out version, and we can seamlessly move in, and we see the data. Eventually, you can see the dendrites here. Eventually, you can see the data at single synapse resolution. So the thing here is that you don't see any unloading or loading. It all happens seamlessly. So another aspect of this work was with the delineations. So mouse brains have, or scientists have calculated some ontologies. So they've organized the brain regions into different areas based on their functionality, for example, sometimes. And because not all brains are the same, every time a mouse brain is imaged, the scientists have to go and delineate the areas to say, this is the hippocampus, this is the cortical plate, and so on. So this ends up being a large set of images. For example, could be 50 images or more. And having a large number of images, or another representation with which is more like polygons, they're not very suitable for real-time applications. So we need to do something better. So the approach that we used for this work was to first inspect the ontology, which is a big tree. And based on how big the tree is and how many children each node has, we can calculate a mapping so that we can use a single identifier, a big number, which is, for example, 64 bits or 128 bits, and pack there the entire path from the root node to the node in question. And each node level can occupy just a few bits. And this ends up being very convenient because we can, if we use this form, we can just use bitwise operations, which are very, very cheap, to do some node comparisons. For example, if two nodes have the same parent, or if one node is a parent of another node, and so on. So I'll skip over some details because we don't have too much time. So the end result is that we can use a single image to store all the masks, and they are stored in a form that it's not very costly in terms of storage, but it's very, very efficient in terms of processing. And for example, here's an overlay of these delineations, and we can hover over, and you can see the different regions, and we can get information, for example, for each of those regions while we're hovering. So the third part is the synaptic puncta. So a large part of the image analysis pipeline is to calculate all the synaptic puncta on the brain. And we might end up having for each of the brain sections, for example, 50 million puncta. And for each of those, we might have up to 17 values. Two of those would be the XY coordinates, so where they are exactly. And the rest of the information is about the intensity of the puncta, the shape, and so on. So they're more circular, more elliptic, etc. So what they used to have, what they used to store this data was a CSV format, which is nice to, there's an existing tool chain and it's convenient, it's human readable, but it's too bulky and too slow to work with in a real-time application. So that wasn't good enough. So we chose a binary format, which just packs everything into a big binary blob. And whereas this is very fast, it's not great because in certain aspects because there's no existing tooling to run statistics and do other processing. So it's non-standard. And what we ended up doing at the end is using HDF5. HDF5 is a very nice and flexible format that allows us, first of all, it's standard. That means we can use existing tool chains to view the data set, view the different puncta. But also it's very flexible in how we store the data inside that structure. And for example, we can choose to not have a hierarchical form, even though that's a major part of the format. And we can also choose to not have any compression because that adds to processing time. So if we do that, we can actually reuse our very simple binary blob and we can just wrap it around an HDF5 header. And what that means that we now have a standardized format, which is HDF5, but what's hiding under there is really like the big data blob. And we can just, when we run the application, we can just copy that data blob as is into memory with zero processing time. So for example, as a comparison to CSV, it would load 15 times faster and it's 30% smaller in terms of size. So that was ended up being very useful. So we have 50 million puncta. The puncta are organized in a Z-order curve. This is, or Morton order, if you've heard that term. And this is a particular ordering of data spatially, and it helps with data locality, which is an important concept when we're doing GPU visualizations. And you can see how it looks like on the image on the top right. And using this Z-ordered data, we can further create a linear quad tree that we can use to reference that data. And linear quad tree is a special optimization structure. And what this quad tree will give us is when we have our rectangle view on the screen while we're watching, while we're viewing the mouse brain, we can very, very quickly identify which nodes of this quad tree are visible on the screen. And the nodes that are visible are the ones that we want to display. And because we don't want to be doing extra work, we do not want to be rendering several millions of puncta that you're never going to see. You only want to put work into what you see on the screen. So that's the entire concept here. And by using this approach, again, it ends up being very efficient to render these points, even though we have millions and millions of them. And here you can see, so this is just a puncta. This is just a puncta view. So what you see is not raw images anymore. This is just small little colored circles. And you can see here, if we zoom in, you can see all the individual puncta. And you can hover over the individual puncta, and it can tell you all the properties of this. So you have a way to identify each individual punctum. And we can also filter puncta so that we can, for example, just see puncta that have a particular intensity range or that are of a particular type and so on. So to help us identify patterns within the data. So the last part is that we have all this data and we have all these forms of viewing the data, and we can run some basic statistics on them. So we can select the regions that we can run statistics over, which could be either the individual tiles that I said in the beginning, or we can do it by ontology node. And the individual tiles you can see on the top right, and the node ones, the ontology ones that you can see on the bottom left. And again, we can use any combination of filters so that we can only view data that we're interested in. And here is an example for how we would use this functionality in that software. So we can visualize the data as heat maps over the tiles, and then we can hover over the individual tile and we can see the actual values. So I want to close with another way of viewing this data. So we've done all this work. We have this very efficient way of viewing the data, so we can do more. For example, we can set up a camera to just explore the data in 3D in the world. And for example, here we can see the hippocampus. And for example, for public engagement purposes, we would convert all this puncta, we would map the individual puncta to flowers. So you can see 50 million flowers, and you can see everything as a big flower field, where the different types of flowers would be the different types of puncta. So this can communicate that the puncta are not just distributed randomly in the brain, but there is actually real structure under it. It might look slightly chaotic, but there is actually structure and organization underneath. I thought since we're already late, I'm done with the presentation. Thank you very much. So I can take any questions. Thank you.
Thank you for your very interesting and nice talk. So any question? Okay.
Thank you, Babs, for the presentation. I have a question how you arrange the tails. So it's clear for me when you zoom in, you move around, you load, enable tails, right? Mm-hmm. But I'm less clear how you can have the overall atlas of the brain, because you have just patches as input, right?
Sorry?
How you can zoom out actually from the images, because you have small patches, right?
Well, we have everything loaded, so everything is in memory. So not everything is in GPU memory, but everything is in CPU in RAM.
Right? Okay.
So when we zoom out and we request different areas, then we check the quad tree and we say, okay, what's visible now? So it identifies the buffer ranges, because we have a big buffer where everything's loaded. And it identifies the buffer ranges that are you talking about the puncta or the microscope image?
The microscope.
All right. Okay. So that uses the sparse visual texture. So this uses some particular graphics technique that I didn't go through here because of time, which is MIP mapping. So while we have the data that are the humongous image, we don't want to always use that, because if you're zoomed out, you cannot see the fine detail, right? So what we have done some pre-processing. So we have this humongous image, and then we create a half-size version of that, reset, and then a half-size version of that, and so on. So it creates an image pyramid. So when we zoom out, we can identify the resolution that we actually need. And we say, okay, we don't need the final resolution data, so let's pick like a smaller level.
Yeah. It's like basically Google Maps. Yeah. So, okay, thank you.
I have one question.
Sure.
You take so many pictures, images, and will it take a long time to process, to mapping those images?
So for pre-processing only? Mm-hmm. Maybe a couple hours, if I remember correctly.
Yeah, I think so, because you have so much data.
Exactly, but the thing is, we put all this effort so that we do that work once. Now, the challenge and what we wanted to do is, when you load the visualization, it loads as fast as possible. That could be, for example, 5 seconds. So in 5 seconds, you have access to this data set, and you can just view everything seamlessly.
Easy to use to analyze the data. Okay. Thank you.
No problem.
Excuse me, we have a question.
Thank you very much for your. And any question in Nanjing side?
Yes, we do. The image we're seeing here, the question is, it's a very nice talk. Give us a lot of light on understanding how we can deal with this data. But the question comes from us is, what is this the data mean anything? This red dots or anything? Is there anything taken from your brain, or is that something you make it up?
Sorry, can you repeat the last part? If the data means anything.
Yes, doesn't mean anything. Is it simulated out, or is it real data taken from the brain, or does it really?
It's real data. So it's the first part shows the raw images, right, taken from the mouse brain, and the puncta, the small little circles that you saw, that is after the image analysis pipeline has run and has identified the synaptic puncta.
Is this puncta mean anything? Is it associated to a neuron point or something?
Sorry, I couldn't hear you.
Does this image point mean anything? Is it related to the brain neuron or any junction or anything? What does this puncta mean?
It has the data is a mouse brain that has been genetically tagged with PSD95 and SAP102. So they show the protein intensity at the time of sacrifice. For the neuroscience part, I will refer you to the two publications because I'm not a neuroscientist myself. Apologies for that.
I see. So there's a way, maybe there's a way to work between you and a real neuroscientist and the data, perhaps. That's excellent work. Thank you.
Professor Liu.
Yeah.
He is from Shufan's group in Napier University. You can connect with her as well.
I see. Thank you. Excellent job. And this data is amazing. It's really amazing. Just that we don't quite, quite understand what does it mean.
He just said the concentration of protein is correct. What the data is the concentration of protein of the mouse brain.
Yeah, but right now it looks like the flowers in the Scotland Highland.
I don't know where you take this picture.
That's the real data. Okay, so this video is the real data using 3D models and some other things that I made. So it's procedurally generated using real data. Yeah.
Great.
So what you see here is the real mouse brain by having applied some color mapping, first of all, for the land. The sky is just an image that I took, and the 3D models, they're kindly donated by some company that makes 3D models, and I just replaced each different punctum type for a different model. So
does the color represent the intensity of the protein?
Yes.
Maybe you can also render it to forest.
Thank you, Charalampos. Great talk.